Okay, welcome everybody to a new global IMMUNOTALK. If you are joining us today for the first time, we would just like to remind you that the goal of this series is to bring to you fantastic immunology in an egalitarian manner and without the need of traveling. Now, of course, we could not do this without our phenomenal speakers. We have today Ron Germain, and I would like to remind you that next week we will have my friend and co-organizer Elina Zuniga presenting. So with this, I will then ask Elina to introduce Ron. We have been having a little bit of internet problems. We hope we will not encounter a problem as the talk proceeds. But if at some point you notice a pause, please do not worry. We will try to solve it, okay? So thank you everybody for joining. And Elina, yeah, it would be great if you can now introduce Ron.
Thank you, Carly. Yes, it is indeed a great pleasure and honor to introduce to you Dr. Ron Germain. Ron was born in New York City in USA, and he has been conducting immunological research since high school. Beginning with the studies of testing whether adult IBEX graft would rescue neonatal animals from graft versus host disease. So it's amazing how early you have started your career in immunology, Ron. So during college, he continued performing immunological research, culminating in the simultaneous award of a master and bachelor degree in just 4 years. And he then received his MD and PhD degrees from Harvard University in 1976. And he continued investigating basic immunobiology, first as the faculty at the Harvard Medical School, and then in the Laboratory of Immunology at NIH, and afterwards and currently as chief of what is now the Laboratory of Immune System Biology at NIH. So the amount and the impact of Ron's contribution to the field of immunology, I think that you will all agree, are truly exceptional. Him and his colleagues have made key contributions to understanding MHC class II molecules' structure-functional relationship, the cell biology of antigen presentation, the molecular basis of T-cell recognition, and the application of system biology as well as computer modeling to understanding immune function. More recently, his laboratory has explored the immune system using dynamic and static in situ microscopic methods that his lab has helped pioneering. And I think we have all been fascinated by the incredible movies of the immune cells that Ron and his team have generated, not only for the pleasure of our eyes, but most importantly, to uncover fundamental new biology of the immune system. So all this amazing research has been published in 400 scholarly research papers and reviews. And there are numerous awards that Ron has received. It's very difficult to pick few because they are so important awards. He has been elected associate foreign members of EMBO and also elected a member of the National Academy of Medicine and National Academy of Sciences in USA. He has also received the Meritorious Career Award, and he has been named Distinguished Fellow by the American Association of Immunologists, among, again, many honors that he has received. So, Ron, it's an honor and pleasure to have you in this seminar series, and so thank you so much for being a global immuno speaker today. I think that Carly, probably we need to help Ron with the microphone. There you go. Okay.
Now I'm allowed to speak. It's good. This is rare that somebody has such control over my ability to speak. So this is a new experience. Thank you for that terrific introduction, and both of you for setting up this really fabulous series. It's been absolutely terrific, especially under the current conditions. So again, I'm really pleased to participate.
And we are delighted to have you. And as always, we want to ask you a question. And today, the question is related to mentorship. And I think that Ron is a terrific person to ask this question about mentorship, because he has been chosen NIAID Outstanding Mentor for training over 70 postdoctoral fellows, including Dr. Cayetano Reyes Sousa and Heiki, who were previous global immuno speakers, and they gave fascinating talks, and many others that now held senior positions in industry or in academia, and they have been very successful. So Ron, based on your amazing mentorship trajectory, we'd like to learn more from you by asking, what is your mentorship style? Was it always like that, or did it evolve over time?
Hopefully, like everything else in life, things evolve, and this is one of those cases where it has. And I'll illustrate it with an anecdote that is very relevant here. So in the very early years of the lab, one of my fellows was Sir Robert Leckler. And Robert said he wanted to have an exit interview, and I didn't even know what an exit interview was, but I said, fine. And he sat down in my office and he said, you know, it's really been terrific being in the lab, but, and the but was, he said, when I show you my data, you tell me the next experiment before I've even thought of it. And maybe that's not an ideal way to train. And I can remember exactly where I was sitting when he said that, because it was very impactful. And it took a few years after that to really internalize what that meant, but the evolution has been toward having the fellows own their own projects, control their projects, and interact with me as a knowledgeable colleague, especially about things besides the narrow questions, the science that, not narrow science, but the specific science they're asking, which they often know more about than I do, but how to do experiments, which controls, the direction to go to put something together as, you know, a package. And so it's very collaborative at this point. And I think, especially for my group, where our aim is to train primarily, if not exclusively, people to be PIs, that ownership early on puts people in the right situation to both test themselves and gain experience in how to run a research program. And so I think that's been the right evolution over time.
That's fantastic. Thank you so much for sharing that. Yeah, I think that that's teamwork, and with the more and more complexity of the science, like the need for more collaborative mentorship. Thank you so much. So we will ask you to then share your screen, because I think that people cannot wait to see your presentation today. And there you go. We can see the slides, and perfect. We can see the slides perfectly well, Ron.
And they're animated, hopefully.
They are animated, yes.
All right, terrific. So I'm going to start with something conceptual, and that is, what's the immune system actually doing? And if you think about either tumors or infectious agents, there are a whole diversity of tumor types, the diversity of infectious agents, and they affect different regions of the body, whether it's the brain, the lungs, skin, liver, digestive tract, or genital tract. And what that raises is this question: how does the immune system protect against diverse, unpredictable disease entities that come at unanticipated times and attack different parts of the body? And so the solution that evolution has come to for this what, when, and where challenge is a combination of soluble effectors, and they're tremendously important. We're in an era where we're worrying about effective vaccines that are largely antibody mediated, for example, but I'm not going to talk much about them today. Mobile elements that can move into the relevant target site to attack either a tumor or an infectious agent, and then organized lymphoid tissues that facilitate the rare cell-cell interactions we need for effective adaptive immunity. And it's the second 2 points that I'm going to concentrate on today. And so just as a very, very brief review for those who are not full-fledged immunologists, we have what are considered primary lymphoid sites, mostly the bone marrow that I'll come back to in a moment, that generate cells, the thymus, where some of those cells mature into functional T lymphocytes, the classical secondary lymphoid organs, the spleen and various lymph nodes, and then the more mucosal associated immune tissue. And then we have the mobile elements. These are, as I say, primarily in mammals generated in the bone marrow. Some emerge as fully mature innate immune cells, such as neutrophils. These can sense inflamed endothelium, adhere, roll, and then transmigrate to mediate effector function in the tissue. And we have the organized lymphoid elements illustrated here as a lymph node, where both B and T lymphocytes enter. They segregate into different locations. And I've asked several thousand people at various talks to tell me why you separate B cells and T cells when they have to get together in the ways that Hai and others have studied in detail. I won't answer that for you. I'll leave it as a question. And if in the so-called questions for me, somebody wants to give me an answer for why you segregate cells that need to interact, that'll be interesting. But in the T zone, the T cells look for their cognate ligands, for their clonotypic receptors. If they're engaged, they can activate, proliferate, differentiate, and then like the innate cells, they can move to find inflamed endothelium and then out into tissues to mediate effector function. And if you think about everything I've just said very quickly, all immune function takes place in either complex lymphoid or a parenchymal tissue context. And therefore, it is impossible to understand immunity just looking at isolated cells or molecules. As important as isolated cell studies are, molecular studies, genetic studies, they cannot tell you how the immune system does its job. We have to look in the tissue and see what's actually happening, meaning we have to image. And the two types of imaging I'll talk about today are dynamic versus static high content imaging. And so there are a couple of questions we want to know answers to. What are the cellular dynamics? And those are both macro and micro scales. So at a macro scale, how long does a T cell have to spend in a lymph node to have a very high probability of finding a dendritic cell with its ligand? And what's the shortest time it can spend there to do that and still get to another lymph node, which is really where its antigen might be? And so evolution has to balance those two rate constants. There's also the question of how long do cells have to stay in contact to transmit information. They don't do it with radio frequency at a distance, even though mediators can be secreted. But for membrane-bound molecules, as shown here, those cells need to touch each other, and they need to touch each other for some period of time, which means they have to stop motion.
Okay, I think we're having
a problem. We'll see.
We'll see if Ron is
Maybe
able to
we send him a chat to see. Okay. Let's see. Let's unmute. Ask to unmute. There you go.
Are we back?
Okay, yeah, let's see, Ron. I think you disconnected. Can you hear us? Okay, there we go.
Can you hear me?
Yes, we can hear you. Yes.
Can you see the key question slide?
Yes.
Okay.
And we could hear that slide. It was the next one, I think, that the one stopped.
Okay. Yeah, let's go ahead. Let's see. Okay.
Hello.
Ron, can you hear us?
I can't hear you. We are trying to connect with him via.
Yeah, so today there has been internet problems. So we'll try to see. Maybe Ron, if you can hear us, I wonder whether we can go to the other possibility of connecting with the phone.
Phone, right?
Yes, let's see. Okay, let's see there, Ron. We are sorry to our participants. And we are grateful for your patience. Let's see. If we encounter problems, we know we are all looking forward to listening to Ron, so we will absolutely proceed to record this at a time when we have a good connection. So we all will have a chance to listen to Ron's wonderful talk. Let's see. Okay, let's see, Ron.
Do you see a pathology slide?
Correct, we do see that. Okay.
All right, so if you're trying to do imaging, there are a number of challenges. We need to worry about resolution, both spatial and temporal. We need to, if we're doing live imaging, disturb physiology in a minimal way. We need to have multiplex detection. If we want to look at many parameters, an observation period long enough to gain useful information. Then we have to process a lot of data, and then we have to display those data. And so I'm going to start with multi-photon dynamic methods. This is what most people are familiar with. They're good for looking at dynamic or temporal issues, but they're only good to up to about 5 parameters, way less than you would look at by flow cytometry, for example. Signaling measurements are difficult. Calcium is pretty much the only one that works in a reproducible manner. You can look for seconds to hours. And other than in some explant situations, this is not good for human studies. It's for mice. So the way you do it is expose the tissue of interest. And so the first thing you have to understand is, except for the ear skin that you don't do surgery on, you do surgery. And you should always be careful to look at whether the people doing this work have told you what controls they run, that they didn't cause unexpected inflammation from doing the surgery. You take a redshifted laser, you collect the plane of imaging information, you focus up and down, get a 3D stack and collect, and then do this over time and play it back about 200 times real speed and with a compression in Z. And you see these movies, for example, T cells moving in the paracortical region of a lymph node, and it looks like they're free to move wherever they would like, but you need to remember they're actually in this environment. They're touching lots of things, and you just didn't make those other lots of things fluorescent so you don't see them. So the lesson here is, don't ever believe what an imager tells you based on what he or she does not see. Always worry about what's in the black. And so I think of the immune system as having the barrier function, which if breached gives you innate responses, and then gradually you develop adaptive. So let's start with innate responses. Here's a sandfly with a successful blood meal from disrupting dermal capillaries. And if we look in the ear of a bitten mouse, we can see a blood vessel over here. We can see a region of damage indicated here, green genetically labeled neutrophils and a few red leishmanial organisms. And if we look at this, we see a lot of things that are useful information. So all the neutrophils come out of the proper side of a blood vessel, so they have accurate targeting over 1 cell diameter. They move very regularly in a straight path, and then they begin to swarm near the damage, but this underestimates the actual information for controlling their movement. If we look here at the side, and we get a free channel here for the dermal collagen, black, as I told you, is not empty space. This is the epidermis. We see the neutrophils going into a particular place, and what they're doing is filling in the bites, and they're reestablishing the barrier function of the tissue until the tissue can heal itself to prevent pathogens from entering the disrupted dermal vasculature and becoming systemic. And so instead of this very simple rolling adhesion transmigration model that most people are familiar with for cell activity in tissues, we have this very complex 3D environment of acellular and cellular components, DAMPs, PAMPs, that these effector cells need to navigate in to mediate their function. And so several years ago, Tim Lammerman, now running his own lab at the Max Planck in Freiburg, went to study this in great detail. He set up what we call a sterile injury model. We use a pulse laser to create a lesion of a fixed size, and we can scale the size with the intensity of the laser pulse. We can avoid clipping vessels, so we know we're not dealing with extravasated platelets at that point, or serum components until inflammation becomes more aggressive. And we know exactly when we start the damage. And when he does that, you see in the oval region, the region of damage. That was to wake you up in case I was droning on. The very directed migration of the red neutrophils, the formation of this tight swarm, and then gradually over a few hours, the accumulation of monocytes. That initiation of the swarming behavior and the directional migration is in concert with the death of the first 1 or 2 neutrophils that touch the damaged area. You're seeing their DNA here from pyridium iodide. These are not necessarily nets, and actually the direction in which you see the DNA move is because of the physical force of squeezing of the generated swarm. And so these cells move from hundreds of microns away in a highly directed manner. And this is largely, but not exclusively, dependent on the leukotriene B4. And so to summarize a lot of work Tim did, you have this damage. There are some neutrophils that come into the region from a variety of attractive signals. When 1 or more of them touch the damage, and it's important, I keep stressing this touch, they die and they start a leukotriene B4 cascade. The LTB4 induces other neutrophils to make the same mediator, and so this spreads very rapidly by signal relay in the tissue. Migration is integrin independent, but swarm formation is integrin dependent, and gradually the monocytes accumulate, and we don't have the data yet, but we think that they're likely to be making the kinds of mediators that Charles Sirhan studies or resolvins that actually counteract the leukotriene to stop things. And we know that that happens because if we image for a few hours, an hour here, we see the motion stop. Now you have to be careful. These animals are anesthetized. They can be cold. They're hypoalimented, and we put a lot of laser power into what we're imaging. And so this could just be an artifact. So how can we tell the difference as to whether this is meaningful physiological cessation of recruitment of innate cells, or this is just a bad prep? And the answer is we can create more damage. If we've tuned the balance between pro and anti-inflammatory mediators to say, you know, enough is enough, and if we imbalance this by creating more damage, we'll shift more toward the pro-activating state, and we ought to see migration again. And in fact, that's precisely what we see. The neutrophils that were in the general tissue area migrate rapidly, and then even some that we've labeled by a photoactivatable GFP in the original swarm are reattracted to the site of the damage. And why is it important to stop this collection of neutrophils? Because if you look at the right side in white, which was the dermal collagen, you see that where you get the neutrophil swarm, you wind up getting a hole in the collagen. So there's collateral tissue damage associated with the neutrophil recruitment, even if that swarm is doing a good job of isolating an infected or damaged tissue site away from the viable or uninfected tissue. And so over the last few years, Stefan Udehard, who's moved back to Erlangen to start his own laboratory, has studied this issue of neutrophil-mediated tissue damage and how that's controlled. And he set this up in a new model system in the peritoneal wall. And the reason we chose this is there's a beautiful population of embryologically derived resident tissue macrophages, the more classical monocyte-derived peritoneal macrophages, and easily recruitable neutrophils in an optically beneficial environment so that we could look at all of these myeloid cells. And so here are the fixed tissue macrophages. They survey the area around them, but they don't migrate. If we add red salmonella bacteria, then we get a massive neutrophil infiltration without the movement of those macrophages. And if we look carefully, some of those neutrophils, when they ingest the bacteria, die, and when they die, we get a swarm. Remember what I told you about death and swarming from Tim's work. And in fact, if we allow the swarms to form and make additional damage in the tissue, just as we did in that slide I showed you, the movie I showed you about going from a resting state back to an activated state, all neutrophils go to where the damage is. And this is the phenotype we've seen in every tissue we've examined, that once the neutrophils are in the tissue, what they're paying attention to is damage, and usually due to signaling from a lead neutrophil that sensed that damage. So what Stefan did is to readapt the sterile injury model to the peritoneal wall. And when he does that, now you see the neutrophils swarm to these corpses in white to digest them. and again disrupt the local tissue structure, just as we saw in the ear. And that raised this question. Cells die in tissues all the time. If that kind of cell death was to lead to this type of neutrophil recruitment and collateral damage, how would you ever maintain organ physiology over time? And so what Stefan did is to say, if we think the cells are intermittently dying, it's probably individual cells. So let's use the laser to kill a single cell. And that's what you see in orange in the middle here. And what he saw when he did that is that the resident tissue macrophages rapidly sense the damage, and then over 10 or 15, 20 minutes, extend processes that completely surround, or as we call it, cloak the damaged cell. And that has a dramatic effect on neutrophil behavior. This is the movie I showed you previously with macro damage. So multiple cells dead. We get this LTB4 dependent streaming from hundreds of microns away. But if we have a single dead cell, although neutrophils arrive, you see that they don't have that directed migration, they don't swarm. And the reason for that is the lead neutrophil can't touch the damage. If it can't touch the damage, it doesn't die. If it doesn't die, it doesn't make LTB4, and therefore there's no swarming. Now, if you think that this is just an artifact of the laser damage, Stefan did perfusion fixed peritoneal walls in completely unmanipulated animals and easily finds, after a bit of looking, these cloaking figures. So this is an ongoing process in tissues all the time. And in tissues that are stressed, so for example, the diaphragm, where you have constant muscle contraction, if you deplete the resident tissue macrophages using CD169 DTR animals and diphtheria toxin treatment, you begin to get necrotic myofibers in association with intense neutrophilic infiltration. And so the lessons from all of this are that the resident macrophages, rather than playing a major role in classical host defense, are actually playing a major role in tissue homeostasis. And they're preventing intermittent minor damage to tissues, leading to this excessive inflammatory damage, but rather allowing healing to occur by local cell replacement, where there's no need for post-inflammation resolution. It turns out that there's an age-dependent transition of these macrophages to a more pro-inflammatory behavior, and that may be associated with what's called inflammaging and maybe the rise in IL-6 levels. And there may be a similar transition induced by amyloid that might contribute to Alzheimer's. These are areas of active investigation. So I want to turn from innate to adaptive responses, and I'm going to skip over the initiation of those responses. And I want to talk about effector function in the tissues. And for most immunologists who are listening, an effector is a dot on a FACS plot that either expresses interferon or IL-17A or IL-4 or IL-9. But most of you are probably not thinking about these questions. When a effector T cell's in a tissue, does it make its cytokine, whatever that is, continuously while it's in the tissue, briefly, or episodically when there's antigen present? What about cell dynamics? Is cytokine delivery local? The cell sees its ligand on a presenting cell and stops, makes cytokine. If it loses that input signal from the TCR, does it now shut off cytokine and move? Or does it get activated and then run throughout the tissue distributing the cytokine? And what about what happens to the T cells? Do they repetitively respond, or do they desensitize from the antigen that's present? And so several years ago, Jackson Egan and Tetsuya Honda studied this process. This is published work, and I'm only going to summarize it very briefly because I think it makes an important point in this era of immunotherapy, which is the model they set up was to transfer TCR transgenic OT2 RAG EGFP labeled T cells specific for ovalbumin into an animal, and then prime the animal with a combination of ovalbumin and keyhole limpid hemocyanin as an irrelevant antigen that would prime the endogenous T cell. We then come back a week later and challenge in the ear only with KLH. That creates an inflammatory environment that attracts effector T cells, including the activated OT2, but they don't see their antigen in the ear until we give the animals peptide intravenously. And this allows us to know within a few minutes window precisely when the T cells we're imaging first see their antigen, so we can address some of the questions that I just raised. And so this is one of the longer movies ever made for intravital imaging. It's 14 hours. And what you see is as soon as we add the peptide, all of the cells basically stop and wiggle. And then over several hours, some of those begin to move again, others move into the field. And then if we give a second dose of the peptide, many, but not all the cells then stop. And so the dynamics look like this. They're moving between 4 and 6 microns per minute in the tissue. When peptide is put in, within a few minutes, they all basically stop, and stop is trackable as sort of 1 to 2 microns per minute movement. And then over 4 to 6 hours, they regain motility. And we and others have shown in the past that T cells basically undergo stop motion when they see a strong cognate ligand. So this stopping is completely expected here, but this regain was unexpected. In terms of the cytokine and answering that first set of questions I raised, there's a monotonic cytokine response. It's off when the effectors are entering the tissue until they see antigen, and then within 1 to 2 hours, they peak in their cytokine response as a cohort, and then as they regain motility, they go back to baseline, which tells us that T cells are making cytokine when they're on their APC, and they're stopping their cytokine production when they begin to move again, and they're not getting an effective cognate antigen generated signal. So it says that the intention is to localize the effector cytokine in the region of antigen recognition. But why are we getting this monotonic response? It could be that there is no antigen left. It's been trogocytosed or somehow degraded or lost. But this is a very fast turnover for that to be happening, given other measurements of how long peptide MHC complexes survive, especially class 2 complexes. And so the other possibility is that T cells are changing their responsiveness. So the way we've looked at that is to do everything I've shown you before: TCR transgenic transfers, double priming of the animal, attraction through a KLH challenge, timed exposure to the cognate antigen, and then we let the cells go through that period of response and then return to migration. And now we transfer more in vitro activated OT2 transgenic cells, but in a different color. And then, as I told you, we always have to worry about the state of the preparation. And so to deal with that, we've put in a third set of cells that are activated, but not looking at any antigen present in the system, in another color to tell us the prep is working well. And when we look at those cells, they're doing what they should. They're moving, as I say, about 6 microns per minute. The ones that had been exposed to their antigen and that had stopped have almost returned to full motility here. They're a little slower, and there are a few that are still stopped. But importantly, a large cohort of the new cells we put in stop, which means the antigen is present, but these cells that had seen their antigen are now beginning to ignore it. That suggests there's something inhibiting their ability to respond to antigen. So we've looked for negative regulatory signals, and there are some obvious candidates. PD1 is a key one, and yes, it rises to a peak precisely at the time that the system begins to shut off its initial response. We don't see that for CTLA4 or LAG3. We've not looked at TIGIT or TIM3 in this early experiment. They haven't lost their T cell receptors or their co-receptors. They do put up PD-L1, which is consistent with them being in a gamma-rich environment in this DTH model. So what does this do to the timing of motility and response? And so this is the control where they have stopped their stop motion, returned to full motility in about 4 hours in this experiment. But if we block the PD-1, PD-L1 interaction with antibody, even at the latest time of the imaging here, 6 hours, we still have a substantial fraction of cells that are not moving very well. And more importantly, they're still making interferon gamma. So this says that PD-1 has a negative feedback role in controlling dynamics and function of effector T cells, not just in chronically infected, so, clone 13, LCMV, not just in tumor-bearing animals, not just in tolerized animals. This is actually a primary regulatory mechanism that deals with normal effector function, not overshooting in the tissue. Just as I said, there's regulation of the neutrophils, the same principle applies here. The immune system as negative feedback controls to limit the possibility of tissue damage. But of course, you want the effectors to do their job. And so how do T cells know whether they're over-responding or not? And they need to track antigen levels, and that's basically being tracked for alpha beta T cells by peptide MHC levels. And if they're constant, then those T cells can, in an anthropomorphic way of thinking about it, say we're doing a pretty good job. The pathogen load is not increasing, so we should begin to shut down, which is what we see here with a non-replicating antigen peptide. Then how do you clear the pathogen, since that's only stasis? Well, if you've got stem-like memory cells that are making new effectors and they come in like the new cohort we transferred, now they're perfectly capable of seeing that level of antigen responding and driving the level of pathogen even lower, and so on. And so it's not a digital kind of ratchet, it's a continuous ratchet where you keep bringing in new cells and they're always able to deal with the ambient level of antigen until it's eliminated, but the system doesn't overshoot and create excessive tissue damage. And so when you talk about exhausted T cells, what you're looking at are cells that have been in a place where they haven't been able to eliminate the antigen and are being pushed into this mode of control without the system being able to fully clear the antigenic load. And so we've used this kind of live imaging over many years in lymph nodes. Mark Bajanov showed years ago that these T cells track along fibroblast reticular cell networks, and I'll come back to the importance of that in a moment. Hai and others in other laboratories have studied the germinal center reaction. Mark Chepa, when he was in the lab, looked at dendritic cell extension from villi in the gut to sample bacteria. We've looked in the kidney. Jackson Egan in particular did many experiments looking at both innate and T cell effector function in mycobacterial granulomas in the liver. With some difficulty, we've done some lung imaging. Other groups have done more. I've shown you the innate response in the ear, and although I didn't show you the movies, those PD-1 experiments involved looking at T cells in both the epidermis and the dermis. We've also looked in bone marrow. I've shown you the peritoneal cavity. So it's a great technique, except we only are looking at a very small volume, even of a mouse lymph node. And so if I warn you about worrying about what's in black here, we don't make fluorescent, then here, everything is in the black. And so we decided we needed to image by histology what we weren't seeing in our 2-photon images to better interpret our data sets. And the problem was when we started doing this, most histochemistry by immunofluorescence was 2, maybe 3 colors. It was red and green, you look for yellow, but everybody else was going down the hall and doing 12 to 15 color flow cytometry, since you needed 3 to 5 markers per cell to distinguish among multiple cell populations you're interested in studying. And that's obviously particularly true in the tumor microenvironment. And so this leads us to discussion of multiplex static methods. There's no dynamics, but as I'll show you, the parameter space can exceed 40 or 50. I'll show you an example over 80. You can look at signaling and post-translational modifications much more easily than we could do in the dynamics. We can look at proteins and RNA under the right conditions, and we can use it with ethically collected human material. And so in our early days, we developed what we call histocytometry. There are other parallel technologies. I'm not going to go through and mention all of them. But I'm only going to give this as an example of the way we've approached it in the lab. And this is work that Michael Gerner did before he moved to start his lab at University of Washington. And what he worked out is the ability to stain in up to 14 parameters at a time, improve the image with a little deconvolution, compensate for channel spillover the way you would do in flow, and then use some software tools to segment cells to create cellular objects with associated fluorescence. But that's essentially flow cytometry data. So we could put our imaging data into software tools like FlowJo, gate and get quantitative information on different subsets of cells. But since we have all the XYZ coordinates for every cell we look at, we could then look at where those gated cells were in the original tissue environment. So that's why it's histocytometry. We're doing everything you would do on flow, but we're doing it with spatial information retained. And I'll show you it works with human samples with antiphosphoprotein and anti-cytokine antibodies, so we can phenotype cells, look at their signaling state, look at their effector function, and position them in the actual tissue, something that none of the single cell methods actually enable you to do. So to give you an example of how this works, if we say that B220 identifies B cells and we make a gate around that cluster, and CD3 identifies T cells, make a gate around that cluster, make this cluster blue, this cluster red, and ask FlowJo to tell us what's going on, we get a perfect sagittal section of a mouse lymph node with peripheral B cell follicles and a central T cell zone. Quantitatively, we get the same answer if we look in a naive SPF mouse lymph node by flow cytometry and histocytometry. That's very satisfying, but this is the important point. If we look at dendritic cells, not lymphocytes, this little tiny wedge you have trouble seeing are the numbers we get from flow unless we go to extreme digestion and extraction conditions. This is what we quantify by histocytometry, and we've validated these numbers. And so the message here is this underrepresentation of certain cell types, primarily myeloid cells and many stromal cells, is true of every method that people use to isolate single cells from a tissue and put into analytical methods like CyTOF, single cell RNA-seq, 10x, et cetera. And we have done this for multiple mouse and human tissues, and this always turns out to be the case. So the single cell data are not what we call ground truth. Actually, the tissue imaging data are more accurate for what the cell populations are that are in that tissue, and you need to correct your single cell data for this extraction artifact. So Michael went and used the markers that existed at the early time to look at the distribution of dendritic cell subsets and found that, as Bernard Mousen had originally reported, for a smaller number of dendritic cell subtypes, they live in different regions with the what we now call CDC1s, primarily deep in the T cell zone, and CDC2s in the more peripheral region. And this goes to a major question. When you look at these cartoons for how adaptive immunity works, there's a real problem. And early in an infection, there might be only 1 in 100 to 1 in a few thousand of the dendritic cells in a lymph node with a reasonable amount of antigen, 1 in 100,000 to 1 million antigen-specific CD8, CD4 cells, or B cells. And when 2 of these cells have to get together, that's already a real problem. And when 3 of them have to get together, a CD4 and a CD8 and a dendritic cell, it cannot be true that it happens at random. And the question is, how does this really happen? And so we published a number of papers on this, so of other laboratories. I just want to highlight a more recent one and then summarize, and this is work that Antonio Batista did in the lab. And what he did is to repeat what Michael had originally shown for CDC1s that are primarily central in the T zone, and CDC2s that are primarily peripheral in the lymph node, and then ask where are CD4s and CD8s. We typically think of them as randomly intermixed in the T zone, but that turns out not to be true. The CD4s have a peripheral bias, whereas the CD8s are more uniformly distributed, or occasionally they're more centrally biased. And so that actually matches the predilection for antigen presentation of the dendritic cells and the T cells. CDC2s are better at class 2 presentation, or more exclusive to class 2 presentation, and that's where the CD4 cells sit. And CDC1s are the ones that can cross-present and are very good for stimulating CD8 T cells, and that's where the CD8 T cells live. And so that's just shown here quantitatively, where we measure the distance of T cells, CD4s or CD8s, from the lymph node center, and see that that parallels the difference for CDC1s and 2s. It's true across mice. It's true for germ-free mice. So this is a general phenomenon. It's actually also true in humans. And the question is, what leads to this? And so we went into ImGen, and there were a few candidates, and the most obvious one was GPR138, which I'll just call EBI2, that Jason Sister has done a lot of work on. It's more highly expressed in CD4s than CD8s. The CD4s and not the CD8s respond to oxysterol ligands in vitro. And if we knock out EBI2, we lose the polarization of the CD4 T cells. And that's true if we also get rid of the oxysterol. What does that mean for function? It turns out that there is a delay in the ability of the T cells, the CD4 T cells, to cluster with their antigen-bearing dendritic cells in the periphery when you don't have EBI2 guiding you to be pre-polarized in that region. And so here at 9 hours, wild-type antigen-specific cells have already made these peripheral clusters, whereas there's a more uniform distribution of the EBI2 deficient cells in that same lymph node. This turns out to discoordinate the ability of the CD4s to help for CD8 T cells. And in this model that Anita Gola worked on in Oxford for her PhD with Adrian Hill, where you prime an animal to adeno-OVA and then come back with nanoparticle IV, you establish tissue-resident memory CD8 T cells in the liver that are highly protective against OVA-bearing malarial sporozoites. And when you look in those animals, if you have a normal animal, it's 80 to 100% protective, but an EBI2 deficient animal, you lose virtually all the protection. Now, the problem here, of course, is maybe there are other things EBI2 does than what I just described. So these experiments were repeated where the only cell that's deficient in EBI2 is a CD4 T cell. And this model, there's no effect of depleting CD4 T cells at the time of challenge, but they are necessary early in order to generate the effector CD8s. And in this condition, when those cells are EBI2 deficient, again, we lose all the protective response. And so there is this non-random distribution of both dendritic cells and T cells in the steady state that they match each other in terms of presentation and response predilection. So it brings responding cells close in the steady state to their primary presenting cells. It's mediated by EBI2. And if you disrupt that, it interferes with the cooperation necessary for a strong cell-mediated immune response. And so this is part of what I was saying before about the non-random way in which the lymph nodes actually work. At the first fractal level, you concentrate antigen-specific lymphocytes and dendritic cells and antigen in one tissue as opposed to diffuse throughout the body parameters. Within the lymph node, there are subregions, as I told you before. In the T cell zone, we have this FRC network. The T cells move on this network, and the reason that's important is the dendritic cells live attached to that network, so you've concentrated the reactants even further and enforce their interaction. We've shown years ago that there are biased walks on this network based on chemokines. Once one T cell interacts with a good dendritic cell, it attracts others in a non-random way, and now I've shown you that there's already a pre-parsing of where the different T cells and the dendritic cells reside to foster their interactions. And it's only with all of these hierarchically organized physical and chemical components that you can take what is inherently an incredibly inefficient system, these very rare cells in the adaptive immune system, and have them rapidly and effectively meet up with each other and generate a response that's fast enough to deal with the pathogen challenge. And so old models in which you think of the lymphoid sites as sort of places where you do pinball behavior of things randomly banging around is not how you get a good adaptive immune response. So that's all wonderful, but that was only up to say 10 parameters or 12 parameters. And you have all the immune cells and their markers, all the epithelial and stromal cells, all the structural elements, tumors. And so over the last few years, we've worked to extend the multiplexity of this approach, and we meaning really Andrea and Evelyn. And they developed a method that we're hopefully going to publish soon called IBEX, in which they do iterative staining in a way that's very rapid and also open source in terms of the antibodies you can use and the colors you can use. And so you stain in 6 to 8 colors, you bleach in a very rapid step. After you do an imaging step, you expose to a new cohort of antibodies, image and iterate this, and the cycles are relatively fast. Depending on how large an area and which microscope you use, they can be as fast as about an hour and a half. They validated several hundred antibodies in mouse and human across many different tissues for doing this. And so this, to my knowledge, is sort of the world's record of doing this type of work. It's 82 antibodies, 65 of which actually react in this human lymph node of several millimeters in dimension. And that then poses a question. What do you do with 65 or 82 parameter data? If you think about the flow work you've done in binary gating, the number of combinations become enormous. And if you want to discover new things where you don't know the path to do the gating, it doesn't really help. And so Nishant Thakura in the lab is a computationalist. What he's done is to work out a machine learning algorithm that compresses all of those channels, 65, 82, whatever it is, to 3, colors them red, green, and blue, and scales the color by intensity, which is one of the benefits of the way we do the staining here. It's not yes and no, it's dim and medium and bright in ways that matter, as we know as immunologists. And this is a reversible algorithm. And so now you can take this multi-parameter stain, where he's showing you 4 colors there, and what he calls rapid, which is his algorithm, recolors it. And I want to stress, the pseudo colors are no longer an individual fluorescent stain. It's the cluster of markers aggregated to define a particular cell type. And I'll show you what that means in a minute, that are illuminated here without segmentation automatically at cellular resolution. And so you see naive B cells outside of the germinal center, defined by 4 markers simultaneously. GCB cells that are in the germinal center and that differ in their location and size and shape to the right and the left, the dark zone and the light zone, with 6 markers. We can identify the dividing cells that are in the dark zone, and we can identify the GCTFH, again, here by 5 markers automatically. And so I won't go into this, but these cluster designations of all these cells that you now can get in an automated way can then be used to look at spatial relationships in the tissue, again, in an automatic manner. We're pretty greedy. We don't think just tissue sections are enough because, again, we have a lot, it's in the black. So we've worked out a new way of doing 3D imaging by clearing in ways in which you can do multiplex staining. The other methods that are out there are very good for looking at 1 or 2 colors, or especially some fluorescent proteins. But if you want to do this kind of multiplex staining, very difficult to do with existing methods. And so here is a mouse lymph node in 5 colors. You see the beautiful branching processes of HEB that you wouldn't recognize if you looked only in a slice, peripheral B cell follicles. But of course, we can do the analysis as if it is in sections throughout all the millions of cells in this multi-millimeter object. And so I want to close very quickly in the last few minutes showing how we've combined all those different tools together with computational tools to ask and I hope answer a very interesting question in biology that could not be addressed without these methods. And so we reported a few years ago that conventional T cells make IL-2. They're surrounded by Tregs that become pSTAT5 positive, and the IL-2 producing cell does not become pSTAT5 positive. So these are the domains in which regulation occurs, but these earlier data did not tell us how regulation actually occurs. How do Tregs actually avert autoimmunity if they're not stopping the conventional cells from becoming activated in the first place, which was the way people had originally thought of what Tregs were doing. And so what Harry has done is to show that instead of IL-2, PD-1 expression marks the endogenous autoreactive cell population. So if you look at this cell now in the side view, it's CD3 positive, it's CD4 positive, and it's PD-1 positive. And we've done these experiments in germ-free mice, in non-gut draining lymph nodes, so they shouldn't have any food antigens. And we see exactly the same number and distribution of these cells. So we're pretty confident they're autoreactive, and these cells are pSTAT5 negative. He then worked out statistical methods to go and look at the interaction of the autoreactive cells with regulatory cells, both in 2D and 3D, together with Kim Young Park and John Sang's lab. And when we do that, it's quite striking. These PD-1 positive cells in the steady state, this is polyclonal repertoire we're talking about, no transgenic transfers, are in very dense microclusters of Tregs in comparison to the PD-1 negative CD4 naive cells that are present. So there's clearly a differential distribution and relationship to the Tregs between these autoreactive cells and non-auto self-responding cells in the lymph nodes. Because we can do many parameters, we can do essentially heat map cluster analysis of the different regulatory cells we see. And the striking thing is that there are two clusters here that have a set of markers that are associated with being effector Tregs, and these are also the pSTAT5 positive regulatory cells that I told you live near the responding conventional autoreactive cells. And we've quantified that here, and you see that it's these effector cells and not the non-effector or resting Tregs that are co-clustered with the PD-1 positive responding T cells. And beside that definition of those nearby cells as effectors, we've done other experiments I'm not showing you that the creation and the clustering of these cells is IL-2 dependent, which says that control of autoreactive cells is not a homeostatic system, it's a negative feedback system. The Tregs are responding to the response of the autoreactive cells. Now, I've mentioned that those cells and their are dependent on IL-2, and their clustering around the conventional cell was also dependent on IL-2, and that led us to try to understand, was that affecting chemosensing or was it doing something else? And that something else was likely proliferation, because these nearby cells are very strongly Ki-67 positive. And then using a confetti system in the way that Caetano did, that he described to you for looking at clonality and dendritic cell precursors in tissue, we're able to show that these local Tregs are proliferating near the autoreactive T cells. And so that now results in a feed-forward response. You're amplifying the number and the suppressive function of the Tregs in response to the response of the conventional cells. It still leaves the question of what are the Tregs doing to prevent autoimmunity? And for that, we did have to turn to transgenic systems, and we used the cell that's specific for a gastric ATPase peptide. And in small numbers of these cells, you do not get disease in a normal biopsy animal. And so we transferred only fewer than 1,000 total cells to wind up with Jenkins numbers, if I can use that term, of 10 or 20 antigen-specific cells in the gastric lymph node. And then the question becomes, in your mind, how are we studying 10 or 20 cells? And the answer is, using our clearing method, we can look at the entire lymph node. All the red objects are the polyclonal populations of Tregs. The few purple objects are this very small number, 10, 15, 20 autoreactive cells, and then we put in some polyclonal cells as controls. And so we can get quantitative and spatially resolved data at essentially physiological levels of these specific cells. And the surprise was that not only do autoreactive cells respond in the presence of Tregs, they proliferate. And over 3 days, they expand 30-fold. But if you keep tracking them, they crash to below our detectable numbers, even using this rather elaborate technique. And so there's many more data associated in Harry's study. I don't have time to go through them. I want to sort of close up things here to say that we've now put this into both a functional as well as a mathematical model, where positive selection of conventional cells is believed to occur at a slightly lower TCR functional capacity with respect to self-ligands. I don't know if I want to call it affinity, avidity, catch bonds. And Tregs are a bit better at responding through their TCR to ambient self-ligands. When they both meet up with dendritic cells, the conventional cells put up a very low amount of CD25 and they make a very marginal amount of IL-2. The Tregs put up much more CD25, and that enables them to preferentially capture the IL-2 made by the conventional cells, depriving those conventional cells of exposure to that cytokine. And normally you would think IL-2, well, you need that for proliferation, but I just told you the cells are proliferating. But we do know from the pSTAT5 and other tests that those cells are not seeing IL-2. So what's happening here? Well, first we have this expansion of the cells to be the eTregs, but what really is going on is something that comes in part from Phil Hodgkin's work. TCR and other input signals can drive proliferation of conventional cells in the absence of IL-2. but they need the IL-2 to survive in that proliferative mode, and that they don't get in the presence of the Tregs, and that now prunes these responding autoreactive cells from the repertoire constantly, preventing autoimmunity. And we've made mathematical models that show that very small changes in CTLA4 or IL-2 receptor expression, or a 40% change in the density of the autoreactive Tregs, will allow the conventional cells to escape. And that's consistent with human data where genetic polymorphisms, EQTLs, lead to autoimmunity. And I'm going to end by saying we're really greedy about our imaging. We want to connect it to genomic data. So we're looking to combine the staining with in situ fish work as shown here. But the really exciting possibility are these chips that Aviv Regev and her collaborators have developed. So at a 2-micron resolution, so subcellular resolution, you can do capture of RNA from a tissue section and then do the equivalent of a 10x-like analysis for gene expression. And we're working with them to pre-stain the tissue section before the RNA capture to get between 10 and 20 markers, so we can directly look at tissue morphology or signaling and relate it to dense transcriptional data sets. Finally, we're also obviously cognizant of the role of the microbiome, and we've worked out ways to also do this kind of RNA analysis for bacteria to lead us to a case where we've got the dynamic data, the histocytometry, and what I called IBEX, the multiplex, which we're now trying to combine into a correlative method where we do the imaging, we know the dynamics of cell behavior, then we stop everything and go to the multiplex static mode to deeply phenotype those cells related to the previous activity. Tools for analyzing complex data, moving this into the 3D environment, and connecting it to genomic work so that we actually can achieve what I said at the outset is important, which is understanding the immune system in the tissue context. Thanks very much.
Thank you so much, Ron. That was excellent. Thanks for your excellent work. Beautiful presentation, despite the initial, minor problems. We could listen to it wonderfully. And special thanks to you and your colleagues for pioneering really these new technologies that allow us to address outstanding questions in immunology. That was wonderful. So before we finish, let me remind you that Ron will be answering questions through Twitter. So there's already a tweet out there. Just find this tweet that says, ask questions to Dr. Ron Germain here. And when you reply, please remember to reply with the hashtag @GlobalImmuno. And I very much look forward also to the next Global ImmunoTalk, next Wednesday, where Elina will be presenting. Now, Elina and I would like to share with you that that is not the last Global ImmunoTalk. We have an updated schedule with the upcoming Global ImmunoTalks until the end of the year. So stay tuned, and hopefully you can join us, and you hopefully can help us spread the word, so all of us interested in immunology can listen to these wonderful, excellent colleagues. So thank you so much. Thank you, Ron, again, for presenting your excellent work, and we'll see you next week.
Thank you for your patience.
That was amazing, Ron. Really incredible, your work. Thank you so much for your efforts to present.
Thank you, and see you next week.
Take care. Bye, everyone.